Okay, we're on. Hello, everybody. This is Rhiju Das. I'm tuning in from Stanford University, along with my lab and several Eterna players and developers here. Here's a view of everybody here, and I want to welcome you to actually the first State of Eterna broadcast for the Eterna project. So 4 years running, and we finally have enough amazing results that we feel like we can have a State of the Eterna discussion. And I want to introduce Adrien. Adrien, can you tune in? And you want to
tell? Hey, Rhiju, and hey, everyone. Really glad to be here, and I wish I could be at Stanford today hanging out with Rhiju and his lab. But as it turns out, my brother has a premiere of his classical music piece today in New York City. So that's why I'm in New York City and joining remotely. But very excited about this State of Eterna broadcast and talk about where we've been and where we're going.
Okay, so I'm coming back in. I'm going to share my screen here. And basically what we want to do today is have two discussions. The first one will be basically Adrien and me reflecting on the history of Eterna, and especially our successes over the last year, and then describing to you why we're so optimistic about the next year. I think this is going to be a critical year, and we'll announce a very I think unique mission that Eterna will hopefully will accomplish over the next year. And the second conversation will happen after these first few slides, where we are going to go into Eterna chat. So at that point, we're going to stop this official broadcast and then go over to the Eterna chat for that discussion. And just a reminder, for those of you who aren't able to make it but are watching this broadcast online on this Friday, January 30th, we'll also have another chat from starting at, I think it's 3:30 p.m. Pacific Standard Time. So hopefully you can tune in if you're catching in later. This is going to be in the style of a conversation, so it's not really a formal address between me and Adrien. And here we go. So Eterna started 4 or 5 years ago with discussions between my lab, which had just begun at Stanford, and Adrien's lab at Carnegie Mellon. And we were just effusing over the amazing discoveries in biology of these intricate machines that used RNA as their main substrate, as their main scaffold, to compute information. And we're carrying out incredibly important biological activities. I'm showing you here on the left the ribosome, which is composed almost entirely of RNA. And it creates all the proteins in all living systems. And it's really, in some sense, one of the most complex machines in existence, and it's been around for 4 billion years. Other machines that you might be familiar with are maybe not so helpful to life, but are antagonistic towards us, like this HIV virus. HIV, the Ebola virus, the flu virus, they're all RNA-based machines that, in this case, attack living organisms. And they carry out intricate computations using their RNA-based genomes. And so these machines have been discovered over the last 2 or 3 decades. But what's really remarkable is their use now in the engineering of biological systems and as eventual therapeutics in disease. And so I'm just going to highlight here a really remarkable advance that occurred really in the last few months. It came out of the Wyss Institute at Harvard, where they took advantage of a recently discovered RNA-based machine called the CRISPR-Cas9 RNP. And there's a schematic of this in the top, but what I want to convey to you is that this is a little complex where if you can introduce it into a cell, it has a guide RNA that'll allow it to go and edit an organism's genome. And it appears to work, it was derived from bacteria, but it appears to work in everything from bacteria to yeast cells to mice and now even humans. And the paper I'm alluding to is called the Gene Drive paper. And what the Church Lab and others have started to show is by using this machine to carry out genome edits, there's even a almost scary science fiction way to send a gene, to create a gene that will then spread throughout actual population in the real world. And so there's this prospect of doing silly things like turning every mouse in the world into a fluorescent mouse, but doing actually very useful things as well, like such as rendering every malaria-carrying mosquito susceptible to, say, caffeine or some other drug that's ubiquitous in the human population. And here, at the heart of this technology, is an RNA-based machine. Now, what's been happening is there's been an explosion of research on reusing nature's RNA machines, whether they're viruses that are used for good or for detecting other viruses, or these so-called CRISPR-Cas9 genome editing devices. But the thing that's a little bit that I think Adrien and I and other players have noticed is that we're reusing these machines, but we're not really making our own from scratch. And I think that's the vision that has been exciting to us. We've seen in other areas of engineering, like such as mechanical engineering, that something remarkable happened when we started. Well, it started in an effort to improve locomotion. Of course, humans started by taking advantage of existing animals that move fast, like horses. But it really took off when we devised our own ways to create our own machines that let us move fast, like sports cars. And now we can essentially move and fly and basically do everything faster than anything that nature has given us. So can we do the same with biological systems? And can we take that understanding to, for example, develop therapeutics based on RNA machines? So that's our driving question. So let me give you an example of an RNA machine that I would love to have. Okay. So I would like to have an RNA that would, for example, detect cancer cells inside a cancer patient, and then for those cells only express a protein that would make them fluorescent, so they'd be easier to see in microsurgery, or perhaps even kill them. Okay, so there's already ways to deliver RNAs into human cells, and for example, to make them fluorescent or to make them apoptose or pass away. So this is a slide from Johan Andresen. And it's a schematic of a piece of RNA that can be introduced into human cells. But I'd be really scared if someone injected this RNA device into me. I would love to instead make sure that there are fail-safes and controls so that it's only active inside cancer cells. For example, there are so-called small RNA or microRNA species that are signatures of tumor cells. So I definitely only want the device to be turned on when this microRNA is present. And I'd also like to be able to turn off this therapy if it or make it dependent on a small molecule, maybe a safe one like caffeine or NutraSweet, aspartame, so that if something goes awry, I could just stop ingesting that small molecule, and then in this case the patient, would no longer be subjected to this therapy. So this is kind of the driving idea between RNA-based therapeutics, and it raises the need for an understanding of how to create little devices that are controlled by small molecules or microRNAs that then can turn on or turn off gene expression. So here's an outline of what that device kind of might look like. I'm showing you, and I'm now framing it in terms of logical terms, and you'll see this is very close to looking like an Eterna puzzle. I'm showing you here in the inputs on the left, there's NutraSweet or aspartame and the microRNA, and when those are both present, we want this device to be active, so that the output, which is shown on the right, will be, in this case, fluorescence of the tumor cell, so that you can see it. But in the absence of the drug and in the absence of the microRNA, or in the absence of either one, we don't want those cells to fluoresce. Okay. Now, you can imagine that people have been creating these kinds of devices. Scientists at Caltech, at Stanford, at Harvard, and many of the labs have been successful in creating proofs of concept of these kinds of two-input devices. But what we'd really like is something that would let us have the device respond to several microRNAs that are signatures of a cell, and to not respond for the microRNAs that are signatures of healthy cells, and perhaps to other small molecules that are signatures of healthy or diseased cells. And so what you really want is a device that can, in this case, take in 4 inputs or more inputs, and then have prescribed outputs. Okay, that's the goal of RNA computation, and it's what we think would be a killer app and a missing application in RNA nanomedicine. Now, just to convey to you what a big challenge this is, there are proofs of concept of this kind of RNA computing, but I can demonstrate to you that the challenge of getting to very complex computers has been unsolved for decades, although it's been recognized. So I'm showing you here a screenshot of the announcement of the Feynman Grant Prize in 1994. This is a still unsolved challenge, and And the challenge involves creating a device that fits within 50 nanometers, and that's very much the scale of the RNAs that we look at in Eterna. But the goal is to make it do a complex computation. In this case, it's sort of a computer sciencey question of creating something that adds 8-bit binary numbers, which can be encoded, for example, in microRNAs. But you can imagine this also as a device for a diagnostic. And in fact, the goal of these kinds of nanotechnology applications is to eventually create devices for nanomedicine. So this has been totally unsolved. And so these kinds of creating these RNA-based machines, folks are doing it. There's a lot of excitement, but we just haven't been able to do it at the scale that we need to start up nanomedicine. So I'm going to let Adrien now step in here to talk about the genesis of Eterna and some recent results.
Thank you, Rhiju. Yeah, so I think that's very inspiring, and it's beautiful to have these open problems to work on. And I think it's worth bearing in mind that although you can draw the analogy between trying to build our own mechanical cars and other sorts of previous feats of human engineering and scientific progress, building these kinds of nanomachines and nanocomputers is really a new frontier for science. And it's going to require really a new kind of science and a new way of doing science. And so in a sense, I think that's why Eterna is so exciting. So one thing that we definitely know about engineering these systems, and Eterna has already demonstrated this, but it's known more broadly in biology, is that we need to do lots of little experiments, and a lot of information needs to be learned and gleaned from each one of these experiments. And it's difficult to do that, and it's different from the way physics progressed and the way other sciences progressed. And so, when Rhiju and I set out to build Eterna, we basically said, is there a way that we can directly involve a very large number of people in these experiments themselves and give them direct access to sort of the underlying substrate of nature, and have them figure out what's going on and tell us that. And so that was really the birth of Eterna, was how do we create a game around scientific experiments themselves. And both because that's an exciting and fun thing to do, but also because this is a new kind of science, and we're going to have to find new ways of doing it. And so, as we go through the Eterna successes, I really think they're not just successes for me and Rhiju, and they're not just successes for all of you, although they are. In a sense, I think that they're successes in pioneering a new way of doing science, and I think that that's really, really exciting and something that I'm just so proud to be part of. So going into those successes, the next slide, Rhiju.
Okay. Oh.
Oops.
Okay.
Oh, that was an animated slide. Okay, sorry.
Okay, I'm going to go to the next one.
Yeah, there we go. Well, so we now have grown to over 80,000 players. We won't say what percentage of those are bots, but I think that we're certainly in the tens of thousands of humans who've played Eterna, which is amazing. 23 millionaires, and then as a recent development, 2 player devs, John Nickel and El Nando, and very proud to have both of you guys aboard, and that's been fantastic, and it's been another sign of just how incredibly special and intelligent and multi-talented the community is, that we can even do this at all. So
I would also argue that it's a first for citizen science movements in general.
Yeah, that's true.
Leaders of the development team are top players themselves.
That is true. And then, of course, one of the really big successes this year was the publication of our paper in PNAS, Proceedings of the National Academy of Sciences. And I'm sure you all know this, but this is actually one of the most prestigious journals in all of science. And I guess just got to say that you guys totally killed it. This is sort of a jaw-dropping result that people can beat all of the can beat all of the sort of major computer algorithms out there at a major and sort of fundamental outstanding problem of RNA design, and then that we were able to train EternaBot based on your observations about RNA folding. I mean, that is so profound. People usually don't understand it. It's very smart people. We talk about Eterna for 2 hours, and they say, "Oh, you know, it'd be really cool if you could sort of figure out what the players were doing and maybe derive some principles from that." And I say, "We did that, and we did it rigorously, and it's in the paper." And they say, "Oh my God, I can't believe it." So this is just I'm so proud of that paper, and I hope that you all are all too. It's really amazing, and it's also been, I think, cited. We just looked it up in Google Scholar about between 20 and 30 times, including in other top journals, which is amazing for a publication that just came out this year. So that's very cool. And then Rhiju and I also published a very interesting paper called Scientific Rigor Through Video Games. Are you going to publish? Oh, there we go. Scientific Rigor Through Video Games, in which we more explicitly laid out this argument that Eterna is not just a trick that we played once. It's perhaps a model for how science should be done by professionals, and in particular by having this remote experimental pipeline, you can essentially avoid some of the most important sort of ethical questions that happen in science when the same person puts forward the hypothesis and does the experiments and so on. And so
Let me just. We just had a slight. We're having a slight issue with the hearing and the muting. But
would you like me to present slides, perhaps?
Here we go. No, I got it. It's on.
Okay.
It's fine.
Okay.
I'm going to mute myself here.
Okay. Let's see here. So I think that I truly do believe that Eterna is both going to today and in the future be not only a source of scientific insight in and of itself, but also a leader in sort of a model of how we can do science and how science must be done, in fact, if we want to solve these grand challenges like the Feynman Prize. And then finally, another sort of super exciting aspect of Eterna is just that it continues to inspire people, not only you guys, the players, but also more broadly. And so Nova did a piece about Eterna, and then they got all excited, and they actually made this RNA Virtual Lab, which is a sort of an alternate version of Eterna with much more involved sort of animated and video description of the RNA science. And so they said that Nova, which is, if you don't know, a very well-known science education organization in the United States, told us that the RNA Virtual Lab was by far their most successful. And we got some really great artists to work on that. So if you haven't played with that, you certainly should. And I think it also indicates, it's an indication of sort of the many sort of deep contributions that Eterna is making, not only scientifically, but also more broadly.
It's also a good time to welcome players who are coming in through Nova Labs. I think we've seen a burst every day of students who have been assigned the Nova Labs by their classes, and then they get hooked on the points into the actual game, and they're trying to make 3,000 points. So welcome to all of you, and to the players who are longtime players, let's make sure to welcome them and help them get into the lab, get 10,000 points.
Yeah, and then Rhiju, do you want to talk about successes in CrowdLab?
Yeah, so the other thing that happened in 2013, 2014 is we had a dramatic expansion in experimental throughput. So this is I think this is basically as profound in biology and bioengineering as the explosion of computers and personal computers were in the 70s and 80s. So what we did was, when we started Eterna, we could only synthesize about 8 to 16 designs per week, and there was a huge amount of competition amongst players to get their designs upvoted so that they could get synthesized. And basically what happened around 2012 is, well, it was getting more and more frustrating, I think, for a lot of us that we couldn't get everything tested, especially when there were so many cool ideas coming out, new motifs being discovered, ideas for looking at natural RNAs within the human body, ideas for creating switch devices, and we couldn't test them all. And we looked around, we realized that with the advent of synthesis and sequencing technologies for RNA and DNA, that those technologies were moving faster than Moore's law in computer science. And Cladwang and I teamed up to take advantage of massively parallel synthesis of DNA and RNA, and to couple it to massively parallel sequencing based machines that let us probe the structure of thousands of RNAs at once. And we launched that with your help in 2013. And on throughout 2014, we've now had a massive number of projects where the ideas are no longer coming so much from the expert scientists, but from the players themselves. And so we're really proud to now say that we've made almost 30,000 sequences that have been designed entirely by players, and they're in almost 400 projects that themselves were designed also by players. Okay, so this is you guys exploring your ideas about zigzag motifs and strategies for making frog leg shapes, and also teaming up with experts like Cody Geary, and Evie Anderson, who are the world's experts in 3D RNA nanotechnology, experts like Todd Lowe at Santa Cruz, who is looking at alternative structures for human tRNAs, as well as, I would say, about a dozen other experts to use the Eterna platform, to use the synthesis puzzles and your own analytic capacity to start getting new results in RNA nanotechnology and RNA folding. And so our big goal, and I think over the next year, is now to take all of these scientific results that we have in the game and to help shape some of them into real publications. So we're hoping to see an explosion over the next year of actual peer-reviewed publications that are marking insights and puzzles that have been solved by Eterna players and then tested experimentally through this new pipeline. So, and along those lines, to help with player publications, we're starting to see some new initiatives that are going to be launched this year. So the first one, and this is actually a player-proposed initiative, is EternaCon. So thanks to all of you who filled out a survey, and thanks especially to Tomo, Muzaki, Vineet, and Machine Elves for sending out feelers to figure out whether we can have a EternaCon convention. So I want to announce that we are officially going to be having an EternaCon. It will be a mini version of EternaCon, where we're going to be trying to take our funds to help fund some players to come out here and have a small-scale convention or conference on June 13th, 2015, here at Stanford University. So stay tuned to hear some more details about it. And if that little convention or conference is successful, we'll then apply for funding to have that as an annual event, and hopefully a growing annual event. And another, I think, very cool initiative, and I think Adrien and I, whenever we have given talks about Eterna, the first question we get is, who's playing your game? Okay? And many players, I think, are curious about who our colleagues are. And of course, we know each other through the chat and in some cases through in-person contacts. But there's an opportunity here with Eterna to ask a bunch of important questions about who is carrying out this new kind of science. Are these players who normally wouldn't have access to experimental resources? Are the players thinking more like expert scientists than like gamers? So these are questions that we'd love to answer. And I'm pleased to announce that starting basically in late February of this month, or this year, 2015, onwards, we're going to be starting a survey series designed by Stanford School of Education researchers called Studying Citizen Science at Eterna. It's going to be purely voluntary for players to take part. But we hope you'll take part and help us understand how our community is special, and then to describe to the rest of the scientific community how they might be able to create these kinds of games and bring experimental resources to a wider community. So that's just a quick summary of successes over the last year. The last thing we want to convey to you is the upshot of several discussions that the development team has had with players and amongst ourselves for what is the next big step for Eterna. And so I'm pleased to announce here what is going to be our mission for the next year. And we've already kind of alluded to it. So we want to now take the next step in setting a foundation for RNA-based nanomedicine. And we think what's going to really capture the attention of the scientific community and the popular press and demonstrate the power of this kind of science is to actually achieve this 5-minute grand prize. So we want to create devices that can carry out computations that will be able to handle up to at least 16 inputs. Okay, and part of the reason that we want to create at least one device that adds binary numbers 8-bit binary numbers is because we want to actually solve something that is widely recognized in the scientific and popular community as being completely unsolved for decades. So how are we going to do this? This is amazing. Well, first of all, the inputs that we're going to be using, they're not just going to be kind of computer science-y, logical inputs, electronic inputs, or chemical inputs. We're going to explicitly be looking at biomedically relevant inputs, where the ones or zeros are going to be the presence or absence of specific RNA species that will eventually we're going to want to detect and take out or sequester in real living cells for therapies. And so I've started a list here of 4 the so-called microRNAs. They're often abbreviated MIR with a number. And these are RNAs that are being transcribed right now in your body. They're very short, about 20-ish nucleotides. And if these microRNAs are being transcribed at the wrong time in, for example, your heart, there is strong signature that you are developing a heart disease, hypertrophic cardiomyopathy, or other things that lead to heart failure. These are RNAs that are being studied in labs worldwide, and explicitly here at the Stanford School of Medicine, both as signatures for the onset of heart disease and as potential targets for treating heart disease. And we're fortunate to have collaborators in those groups. Now, so you're going to see these specific sequences showing up over the next few months. And what are we going to do with it? Well, we want to create other RNAs that sense when those other microRNAs are present. So the first target here is to diagnose when a cell is expressing, for example, the first 2 of these microRNAs, but not the other 2. Okay? And we want to do this in various different kinds of combinations to create little diagnosing sensors that we can use to test if a player's blood is creating these has these microRNAs, and also we'll use these devices within cell culture studies to understand the mechanism by which these microRNAs are leading to heart disease. We want to create devices that are have even more complex computations, for example, that will count how many of these microRNAs are present. You can imagine how you'd love to have a sensor that says, okay, really quickly, 3 of these 4 microRNAs are present. You're hitting the danger level. Okay? And that basic counting is also one of the basic things you need to do with computers. And there's going to be a set of puzzles coming out over the next year. And then finally, we're going to be trying to move towards solving this Feynman prize, where we want to create arithmetic logic units that will let us add up the numbers of, for example, microRNAs or other molecules in the body. And here, the explicit first challenge is to create a device that adds 2 2-bit numbers, okay, where the digits are encoded by the presence or absence of each microRNA. Okay. Now, how are we going to do this? Okay, and that's a step along the way to getting to adding 2 8-bit numbers, which is the Feynman prize. So how are we going to do this? You're going to start seeing a whole bunch of new game features that are going to be coming out over the next year. So this includes things like multiple input puzzles. We're going to have to be able to, we want to sense 2 to 4 microRNA inputs. Our puzzles have to encode 2 to 4 or many different states that involve all the combination of those inputs. So I'm showing you here the new puzzle. It's actually not yet deployed within the game, but Nando has set it up. And you can see, unlike our standard switch puzzles, it's got not 2 states, but 4 states. Okay. And we're going to ask the question, can players grok this system? Can they actually solve these kinds of puzzles that involve more than 2 states? This is an unknown question, but we're quite confident that you can get to these larger puzzles. And the other thing that we're going to be launching over the next year are new kinds of experiments. So we can currently do 1,000 designs a month using this single nucleotide resolution shape mapping or chemical mapping. But I think many of you have started to see that we are developing a new experimental strategy that will let us target 100,000 designs a month, perhaps in combination with about 100 to 1,000 different kinds of inputs each. And that's using a technology developed literally in the last year by Will Greenleaf's lab, and that is being advanced by Johan Andreasen. And we weren't sure whether the current community could create designs at this scale, 100,000 designs per round. But we were pleasantly surprised when earlier this month, in January, we had an explicit sprint where we wanted to get 6,000 designs to stress test this new platform. And we found that players, within a matter of 2 weeks, didn't just give us 6,000 designs, but gave us, I think, more than 10,000 designs, 12,000 designs. And we're now synthesizing all of them to see if we can experimentally characterize them. So that's just super exciting. And let me just give you a sense of timing here. Our goal is to get to 400-bit computations. That is the counting, diagnosing, and adding devices. We were hoping to get there by the summer of this year. Okay, we want to discuss it with the players who attend EternaCon Mini, and then decide at that point. Well, are we going to need more players, or can we do it with the current set of puzzles and the current set of players? Are we going to need completely new features for the game? So that's going to be this really important decision point. And if we decide that the answer is yes, we can do it, then our goal is by 2016, that is by over the next year, to create 16 input devices that can handle 16 inputs and to go for this Feynman prize. So that's the mission. And we are incredibly excited about trying to achieve it with you. In closing the State of Eterna address, I want to end by giving a round of applause, first of all, to the developer team. So I'm going to there are folks who have been with the project who are now alumni, and let's highlight their accomplishments. Justin Kwong and Diana Seo were with the project for several years each. Justin launched EternaScript and was a critical developer of the Cloud Lab project system. And Diana, or both Hyung Seo, created the new sequence, the sequence viewer and the lab tracker. Ayush Dara from Berkeley helped with the Nova port. And Ji-Hyun Lee, I think almost all of you know, was the creator of the Eterna game. And so we congratulate them on their major contributions to Eterna, and we're actually looking forward to G's new game, which will be coming out in the next few months. It's not a scientific game, but it's going to be beautiful, I'm sure. And then we want to congratulate the current team, especially several new members. So first of all, Anne Cladwang has been carrying out essentially every month the Cloud Lab experiments and has not failed yet. And in fact, is literally right now has transcribed the RNAs for the current round. Johan Andreasen is a new member of the team who's piloting this amazing technology, the RNA array. Mike Bao joined our team to work on the Nova port, and you'll be seeing, I think at the end of February or early March, the deployment of a tutorial system where we're going to let players create their own tutorial puzzles that they can share with other players. So that's going to be a really important advance. I'd like to welcome Joe Yesselman and Vineet Kosaraju. They piloted the Eterna 3D game last summer, and so you stay tuned. You'll hear more about that initiative over the next year or two. Michelle Wu is a new person. You may not recognize her name, but you've almost certainly recognized her creation, which is an advanced version of EternaBot that can solve switches. And so she's a rotation student with the Eterna project. And finally, and perhaps most importantly, we'd like to welcome John Nicole, who has basically gone and fixed nearly every aspect of Eterna over the last year. He is our I would say, lead developer now. We want to congratulate him and then the newest member, the newest player developer is Nando, Nando888, and he joined basically in the last month, but has already deployed about 10 incredibly critical features for getting us to our new mission of creating complex computers to win the Feynman Prize and to set a foundation for nanomedicine. And then finally, we want to thank all of you. We can't thank all the players, obviously. There's 80,000 of you, one by one, but we want to thank you in aggregate. And we hope that, thank you for tuning into this, and we hope that you keep playing and stick with us for the chat. So we're now going to go offline on the broadcast. We're going to do the chat online. And let's see, it's about 10 a.m. Pacific Standard Time.
Rhiju, would it make sense to some of the devs, are they there with you at Stanford? Do you want to introduce them in person, or?
Great idea. Yeah. So let me see who's here. Joe and Michelle are here, and I think John and Nando are tuned into chat. Let me just at least. Would you guys mind being? Was that embarrassing?
Sorry, I didn't mean to put you guys on the spot.
They might be embarrassed.
Oh no!
Let me just get the. Okay. Let me just show you. Okay, over here, Michelle, can you raise your hands? Okay, Michelle is the AKA Eterna bot. And Johan, you want to raise? Yeah. So that's Johan Andreasen. And then over here, let's see. Does Anne raise your hand? All right, give a round of applause for Anne. And then there are several folks here who are players and not quite active developers. And then over here is the mighty Joe Yesselman, who's been with our broadcast and is the co-creator of Eterna 3D. Okay, so we're gonna tune out now. Adrien, you have anything to add?
No, I am inspired, Rhiju.
Okay, let's go. Okay, so hopefully the players are too, and we can have a really fun dev chat for the next half hour.
Okay, so
okay. And also, folks who are watching this on the YouTube broadcast, we're going to have another dev chat at 3 p.m. Pacific Standard Time, at 6 p.m. Eastern Standard Time. So that's later today on January 30th. So tune into that if you aren't tuning into the live broadcast. All right.
But I will not be able to make that one because
I'll be there.
Yeah. Okay.
Okay. All right.